Hey everybody, and welcome back. So today we're going to be looking at our next lab, and I'm going to try to do a little bit of an intro similar to what I would have done in class if we were to all be meeting, as we look at Lab 7, which is Chapter 8 in the lab manual, Early Paleozoic Life. Okay, so just a brief introduction to this lab before you all get started on going through the exercise in Canvas. So this lab is going to be focusing on the early Paleozoic, the life of the early Paleozoic. So just for example, here's the Middle Ordovician, which is a nice representation of generally what things were like in the mid-Paleozoic or early Paleozoic. Notice that Laurentia, or in Central North America, is straddling the equator and rotated about 90 degrees, where our current east coast would have been our south coast at the time. So as we've talked about before with our slough sequences, these mega sequences, we're at a general high sea level time. So yes, there are ups and downs like any time with sea level. But if you look at the Cambrian, Ordovician, and Silurian periods and what sea level is like during this time, this is when we have the Sauk and Tippecanoe sequences. And our regressions in between the Sauk and Tippecanoe is pretty minor. So generally speaking, this is a time of very, very high sea level. So what we want to think about as we're going through this exercise is what type of evolutionary adaptations do we see in marine organisms at a time when sea levels are going up, up, and up. So I know in lecture I've mentioned before, I think before everything kind of went strange when we were still meeting in class, that the Linnaean classification system is kind of being phased out in favor of something like the cladistics classification system. And while that is true, the phylogenetic system that we'll be looking at here with the Linnaean system, the kingdom, phylum, class, order, and so on, is still something that we use to some degree. So we'll be following along with that, but don't worry, I'm not going to be asking you to identify things by their species or genus name, but I do want you to know the classification of some of these organisms. So we're going to start out with kingdom Animalia. In fact, today all we're looking at are animals because it's most of the early Paleozoic, that's all we have. We don't have a lot of plants yet. We'll talk more about plants in the late Paleozoic. So of kingdom Animalia, the first phylum, subdivision of animals that we'll be talking about is Cnidaria. Now in your lab manual, this is sometimes called Coelenterata. The more current proper name for it, though, is Cnidaria. And there's numerous different subdivisions of Cnidarians into different classes. The one we're going to be focusing on is Anthozoa, because Anthozoans make up a majority of Cnidarians. And Cnidarians include corals, sea anemones, Portuguese man-o'-war, and also jellyfish. So the two types of anthozoans that we'll be looking at today are corals, two orders of corals, the tabulates and the rugosans. Now we've talked a little bit about these in the lecture videos that I've done, but just to kind of recap, if you were to go scuba diving in a reef today, you wouldn't see any of these because they're all extinct now. All of the modern day anthozoan cnidarians we have are Scleractinian, a totally separate group or order of anthozoans that we'll talk about later on. So we have the two orders, the tabulates and the rugosans. At the top of this image, you see solitary and colonial rugose corals. Notice how they're characterized by having radiating septa, these walls that kind of go from the outside of the coral inward. And this allowed the tentacled, stinging-celled polyp animal that actually makes up a coral living part to kind of grip inside. So the way that coral animals or polyps actually feed today is they sit around and wait for something to swim by and they zap it with this stinging cell. This is why you should never hug a coral with this venomous stinging cell, and it'll then stun it and then it ingests it. So with our rugose corals, they have solitary and colonial versions. The tabulate corals that you see on the bottom are all colonial and they're much, much smaller. So for example, if you look at the colonial tabulate coral, Halocytes there on the left, the big wide open spaces in between that chain looking structure, that's not where they were living. Look at the tiny little pores on the little chain link itself. That's where the polyps were actually living. Similarly in Phagocytes, those weird little pentameral and hexagonal discs you see, those are columns and each one would have a series of those living in it. So in each of these examples of tabulate corals, you might have had 10,000 different animals of the same species living in there. It's like a condo. With the colonial and the solitary, anywhere between 1 to maybe a few hundred. So very different types of corals, but both major reef contributors in the early Paleozoic. Our next phylum is phylum Mollusca. Now, mollusks are one of the most diverse groups of animals alive today. They include numerous classes, including gastropods, which are all of the snails, bivalves, which are all of our clams and oysters, and then this group, cephalopods, which today includes squid and octopus. Nautiloids are a very specific kind of cephalopod that today is only represented by one type, and that's the chambered nautilus, which you see here. And while it's the only cephalopod alive today with a shell, think squid and octopus don't have a shell, our Paleozoic nautiloids were the most abundant type of cephalopod, and they were really, really big. Like some of them, the straight-shelled nautiloids, like you see at the bottom of the screen, could be up to 12 feet long. And these were the dominant predators living in the early Paleozoic seas and oceans, including here in Wisconsin. So when we look at that Ordovician limestone that makes up our bedrock, we find a lot of these big nautiloids in there. The other really abundant phylum of life alive today on the planet are arthropods. There's more species of arthropods than anything else on the planet combined. So it's really a bug's world. But we're going to be looking at a very specific class of arthropods, and these are trilobites. So arthropods in general are your crustaceans and insects and arachnids and relatives. Trilobites are a very specific group, and of course, they're today all extinct. But what makes them unique is that they were very, very abundant throughout the Paleozoic, and their shells are actually made of calcium carbonate, so they actually make really good fossils. Things like cockroaches and flies and ants usually don't, because if you step on them, they go crunch. If you take a shrimp, you can peel it. That's made of chitin. These had that calcium carbonate shell. So when we look at the body of a trilobite, there are two different ways we can identify them. We can talk about their latitudinal divisions and their longitudinal divisions. The latitudinal ones, the cephalon, thorax, and pygidium, that's the head, body, and tail of the trilobite. Okay. The longitudinal divisions would be the axial lobe and then the left and right pleural lobes going from top to bottom or front to back. And that's actually where we get the word trilobite, 3 lobes. Another structure you'll see on the cephalon is this kind of curved structure around the face, and these are called the facial sutures. Arthropods grow by molting. When they get too big for their exoskeleton, they have to leave that exoskeleton, grow, and then mineralize a new one. And so they're doing this by breaking parts of their face apart, and that's what these very faint lines are that run around the eyes, are their sutures, the facial sutures, which allow them to break apart, leave, and then molt, and then grow. So we use these different features to help us identify different trilobites. So for example, the large image of the trilobite you see on the left, kind of on right hand of the screen, but to the left, is Elrathia. This is a very common Cambrian trilobite. And you'll notice that its cephalon has 2 little kind of points at the edge, right at the very tips of it. Now, if you look at the 2 trilobites to the left of that, those are phacopoid trilobites. At the top, you have phacops, and the bottom, you have Flexicolemini. You don't need to know these names, but they have different abilities. They can curl up into a ball for protection, which is both fascinating and, let's be honest, kind of adorable. But they have a very different structure on their cephalon. They have a large raised central area we call a glabella. They don't have the spines. They have fewer segments in their thorax. These are all the different ways that we would describe like what's going on in the axial lobe of the cephalon or the left and right pleural lobes of the pygidium. We use these as coordinates to help us identify different parts of trilobites. Now, the next 2 groups that we're going to talk about belong to a common superphylum. So kind of would fall in between kingdom and phylum, would be superphylum, or maybe subkingdom, right? And this is Lophophorata. Both of these types of organisms have a lophophore, which is a filter feeding apparatus that they use to filter water out as they're breathing and eating, and they'll ingest any organic materials that get stuck in their filter or their lophophore. And lophophores look weird. They almost look like a long coiled feather that they extend out and then filter feed. Our first phylum of lophophorates is the brachiopods. If you were to go to a beach today and pick up shells, you'd be picking up bivalve mollusks. Back in the early Paleozoic, you'd be picking up the shells of brachiopods. So they are as different from bivalve clams as we are from bivalve clams. We're all separate phyla. There are 2 classes of brachiopods. There's the inarticulates, which we'll be looking at mostly today, and then the articulates, which we'll look at more next time. Now, when you look at the inarticulate brachiopod on the screen, that's actually a modern lingula, a modern inarticulate brachiopod. The cool thing is these things haven't changed much in 550 million years. They were one of the first brachiopods, and they're still around today. The articulates became very, very abundant in the middle to late Paleozoic. Most of them are extinct now. Now, how do you tell the difference between a bivalve and a brachiopod? Because they superficially look different. And that's because brachiopods are kind of like bivalve mimics. Or maybe it's the other way around. They look similar because they do similar things, but they're quite different. One of the good ways of identifying the differences are their planes of symmetry. So if you look at a brachiopod, where could you cut that brachiopod to make it have mirror images of itself? And really, it's through the center of both shells. So the hinge is going, let's say, left to right. The plane of symmetry is perpendicular to that. With bivalves, which you can see there to the right, bivalves have a hinge, and the plane of symmetry is parallel to the hinge, but each shell itself is asymmetrical. So in brachiopods, the two shells look different, but they're both symmetrical, have symmetry. And in bivalves, the two shells are symmetrical to each other, but neither one has symmetry in and of itself. Okay. In both cases, they sit there and they filter feed, but brachiopods do it with a lophophore. So in the image on the upper right of the screen, you have what looks kind of like a little man-eating plant or Venus flytrap thing. That's actually a brachiopod. They use that fleshy stalk known as a pedicle to burrow down in the sediment. They open the brachial and pedicular valve, and they extend out their filter feeding apparatus, the lophophore, and they breathe and eat at the same time. Now, our second type of lophophorate is phylum Bryozoa. Again, bryozoans are still around today, but much lower in diversity than they were in the Paleozoic. And just while brachiopods were kind of copycats of bivalves, bryozoans are copycats of corals. They function very similar. They look similar. They're colonial. This is a stick bryozoan, also known as a halopora, and each little cavity you see, little hole on this bryozoan, is where an individual animal, zoid, was living, a little individual bryozoan animal. And they have tiny little lophophores that they extend out and filter feed. So even though they look kind of like corals and they're behaving kind of like corals, they're a totally separate phyla, just as bivalves are a separate phyla from brachiopods. We'll be checking in next week with superphylum Lophophorata, especially to see how these two types of phyla have changed as the planet changes throughout the Paleozoic. Okay, so now as you begin Lab 7 on early Paleozoic life, there's a couple of things I want you to think about. I want you to please make sure you are reading the lab manual. I know it's a little different than what I've put together on Canvas, but that lab manual, hey, you've already purchased it, you might as well use it, is going to have some really important stuff in there that I can't just easily replicate on Canvas. I still want you to complete the pre-lab assignment because those are more critical thinking questions about understanding the reading that comes before the lab exercise. So I still want you to read through that lab exercise, especially the intro part, just as you would if we were meeting in class. So look at Appendix B on page 195. That's really, really going to help you with the next 4 labs. This one, late Paleozoic, and the last 2 that we're going to do for the semester. Also, the study guide on page 292 is to prepare you for the last quiz. Not the last quiz, the next quiz. It won't be exactly the same as the format I was originally planning for the quiz in this class. It was going to be very similar to the rock and mineral quizzes where I'd pass fossils around. But I've been able to put a quiz together, I think, that replicates that as best as we can. So make sure you are still using that study guide on page 292. Okay, so I've taken the lab manual, I've gone through the questions, and I've put on Canvas in an online format something very similar, but with some differences. So obviously you want to be answering the questions on Canvas, but I want you to try to read through that lab and look at the questions in there too. Maybe the wording might help you out a little bit. And finally, when you log into Canvas to do this, the labs are set up as quizzes, but I'm not going to be grading you like a quiz. They're going to be graded the same way you would in lab. So it's more about completing it and making those proper corrections. Okay, so I'm grading you on the completion and the effort, not whether you get things right or wrong. So don't worry about that. I only have a few options when I'm putting things together in Canvas for assignments and quizzes. So if it says quiz, yet it's a lab exercise, don't worry about it. You're going to be graded the same way. It's still going to be worth, same kind of number of points as you would before. So until next time, make sure you're keeping up with the videos and the readings and the labs. If you have any questions, please feel free to reach out to me, and we'll talk to you all next time. Hope everybody's doing well.